Chapter 4 Principles of Inheritance and Variation. Have you ever wondered why an elephant always gives birth only to a baby elephant and not some other animal? Or why mango seeds form only a mango plant and not any other plant? Given that they do, are the offspring identical to their parents? Or do they show differences in some of their characteristics? Have you ever wondered why siblings sometimes look so similar to each other or sometimes even so different? These and several related questions are dealt with scientifically in a branch of biology known as genetics. This subject deals with the inheritance as well as the variation of characters from parents to offsprings. Inheritance is the process by which characters are passed on from parent to progeny. It is the basis of heredity. Variation is the degree by which progeny differ from their parents. Humans knew from as early as 8000 to 1000 BC that one of the causes of variation was hidden in sexual reproduction. They exploited the variations that were naturally present in the wild populations of plants and animals to selectively breed and select for organisms that possessed desirable characters. For example, through artificial selection and demonstration from ancestral wild cows, we have well-known Indian breeds. Example, Sahiwal cows in Punjab. We must, however, recognize that our ancestors knew about the inheritance of characters and variation. They had very little idea about the scientific basis of these phenomena. 4.1 Mendel's Law of Inheritance. It was during the mid-19th century that headway was made in the understanding of inheritance. Gregor Mendel conducted hybridization experiments on the garden peas for 7 years for the period of 1856 to 1863 and proposed the laws of inheritance in living organisms. During Mendel's investigations into inheritance patterns, it was for the first time that the statistical analysis and mathematical logic were applied to problems in biology. His experiments had a large sampling size which gave greater credibility to the data that he collected. Also, the confirmation of his inferences from experiments on successive generations of his test plants proved that his results pointed to general rules of inheritance rather than being unsubstantiable ideas. Mendel investigated characters in the garden pea plant that were manifested as two offspring traits. Example, tall or drop plants, yellow or green seeds. This allowed him to set up a basic framework of rules governing inheritance which was expanded on by later scientists to account for all the diverse natural observations and the complexity inherent in them. Mendel conducted such artificial pollination or cross-pollination experiments using several true breeding pea lines. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance and expression for several generations. Mendel selected 14 true breeding pea plant varieties as pairs which were similar except for one character with contrasting traits. Some of the contrasting traits selected were smooth or wrinkled seeds, yellow or green seeds, inflated that is full or constricted green or yellow pods, and tall or dark plants. Table 4.1 shows contrasting traits studied by Mendel in pea, characters and contrasting traits. Stem height is tall or dwarf, flower color is violet or white, flower position is axial or terminal, pod shape is inflated or constricted, pod color is green or yellow, seed shape is round or wrinkled, seed color is yellow or green. 4.2 Inheritance of one gene. Let us take the example of one such hybridization experiment carried out by Mendel where he crossed tall and drop pea plants to study the inheritance of one gene. He collected the seeds produced as a result of this cross and grew them to generate plants of first hybrid generation. This generation is also called the filial progeny, filial 1 progeny or the F1. Mendel observed that all the F1 progeny plants were tall like one of its parents. None were drop. He made similar observations for the other pairs of traits. He found that the F1 always resembled either one of the parents and that the trait of the other parent was not seen in them. Mendel then self-pollinated the tall F1 plants and to his surprise found that in the filial 2 generation some of the offspring were dropped. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dropped were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. The tall and drop traits were identical to their parental type and did not show any blending. That is, all offsprings were either tall or drop, none were of in-between height. Similar results were obtained with the other traits that he studied. Only one of the parental trait was expressed in F1 generation, while at the F2 stage, both the traits were expressed in the proportion 3:1. The contrasting traits did not show any blending either at F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down unchanged from parent to offspring through the gametes over successive generations. He called these things R factors. Now we call them as genes. Genes therefore are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different from the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage and the small alphabet for the other trait. For example, in case of character of height, capital T is used for the tall trait and small t for the drop. And capital T and small t are alleles of each other. Hence, in plants, the pair of alleles for height would be capital T, capital T, capital T, small t or small t, small t. Mendel also proposed that in a true breeding, tall or drop P variety, the allele pairs of genes for height are identical or homozygous. Capital T and small t respectively. Capital T and small t are called the genotype of the plant, while the descriptive terms tall and drop are the phenotype. What then would be the phenotype of a plant that had a genotype capital T small t? As Mendel found that phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance. He proposed that in a pair of dissimilar factors, one dominates the other, as in the F1, and hence is called the dominant factor while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dropness, that is recessive. He observed identical behavior for all the other characters or trait pairs that he studied. It is convenient and logical to use the capital and lower case of an alphabetical symbol to remember his concept of dominance and recessiveness. Do not use capital T for tall and small d for drop, because you will find it difficult to remember whether capital T and small d are alleles of the same gene or character or not. Alleles can be similar as in the case of homozygote capital T and small t, or can be dissimilar as in the case of heterozygote capital T small t. Since the capital T small t plant is a heterozygous for genes controlling one character, that is height, it is a monohybrid, and the cross between capital T and small t is a monohybrid cross. From the observations that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and drop plant produce gametes, by the process of meiosis, the alleles of the parental pair separate or segregate from each other and only one allele is transmitted to a gamete. This segregation of allele is a random process, and so there is a 50% chance of gamete containing either allele, as has been verified by the results of the crossing. In this way, the gametes of the tall capital T plants have the allele capital T, and the gametes of the drop small t plants have the allele small t. During fertilization, the two alleles, capital T from one parent, say, through the pollen, and small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T small t, Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of zygotes, the F1 and F2 plants can be understood from a diagram called Punnett square as shown in figure 4.4. It was developed by British geneticist Reginald C. Punnett. It is a graphical representation to calculate the probability of all possible genotypes of offspring in a genetics cross. The possible gametes are written on two sides, usually the top row and the left columns. All possible combinations are represented in boxes below in the squares, which generates a square output form. The Punnett square shows the parallel tall T male and drop t female plants, the gametes produced by them, and the F1 T progeny. The F1 plants of genotype T are self-pollinated. The symbols male and female are used to denote the female eggs and male pollen of the F1 generation respectively. The F1 plant of the genotype T when self-pollinated produces gametes of the genotype T and t in equal proportion. When fertilization takes place, the pollen grains of genotype T have a 50% chance to pollinate eggs of the genotype T as well as of genotype t. Also, pollen grains of genotype t have a 50% chance of pollinating eggs of genotype T as well as of genotype t. As a result of random fertilization, the resultant zygote can be genotypes T, T or t. From the Punnett square, it is easily seen that 1/4 of the random fertilization lead to T, half lead to T, and 1/4 to t. Though the F1 have a genotype of T, but the phenotypic character seen in tall. At F2, 3/4 of the plants are tall, where some of them are T, while others are T. Externally, it is not possible to distinguish between the plants with the genotype T and T. Hence, within the genotypic pair T, only one character T tall is expressed. Hence, the character T or tall is said to be dominant over the other allele t or drop character. It is thus due to this dominance of one character over the other that all the F1 are tall through the genotype is T. And in the F2, 3/4 of the plants are tall through genotypically, half are T, and only 1/4 are T. This leads to a phenotypic ratio of 3/4 tall, that is 1/4 T plus 1/2 T, and 1/4 t, that is a 3:4 ratio, but a genotypic ratio of 1:2:1. The 1/4:1/2:1/4 ratio of tall as to heterozygous as to dwarf is mathematically condensable to the form of a binomial expression, that is ax plus by bracket square, that has the gametes bearing genes T or t in equal frequency of half. The expression is expanded as given below. 1/2t plus 1/2t bracket square is equal to 1/2t plus 1/2t into 1/2t plus 1/2t is equal to 1/4 T plus 1/2 T plus 1/4 t. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarf was homozygous, that is t. What do you think he would have got had he self-pollinated a tall F2 plant? From preceding paragraph, it is clear that though the genotypic ratio can be calculated using mathematical probability, by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 has T or T composition cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism, that is pea plants here showing a dominant phenotype and whose genotype is to be determined, is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. Figure 4.5 shows the results of typical test cross where violet color flower capital W is dominant over white color flower small w. Using Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get using the genotype of this cross? Can you give a general definition for a test cross? Figure 4.5 shows a diagrammatic representation of a test cross. Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance. The first law or law of dominance and the second law or law of segregation. 4.2.1 Law of dominance. In that, first, characters are controlled by discrete units called factors. Second, factors occur in pairs. And third, in a dissimilar pair of factors, one member of the pair dominates, that is dominant, the other is recessive. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 and the expression of both in the F2. It also explains the proportion of 3:1 obtained at the F2. 4.2.2 Law of segregation. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 stage. Though the parents contain two alleles during gamete formation, the factors or alleles of a pair segregates from each other such that a gamete receives only one of these two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each one having one allele with equal proportion. 4.2.2.1 Incomplete dominance. When experiments on peas were repeated using other traits in other plants, it was found that sometimes the F1 had a phenotype that did not resemble either of the two parents and was in between the two. The inheritance of a flower color in the dog flower, that is snapdragon or antirrhinum species, is a good example to understand incomplete dominance. In a cross between true breeding red colored and true breeding white flowered plants, the F1 was pink as shown in figure 4.6. When F1 was self-pollinated, the F2 resulted in the following ratio: 1 red, 2 pink, 1 white. Here the genotype ratios were exactly as we would expect in the Mendelian monohybrid cross, but the phenotype ratios had changed from the 3:1, that is dominant, as to recessive ratio. What happened was that capital R was not completely dominant over small r, and this made it possible to distinguish capital R small r as pink from capital R red and small r white. Explanation of the concept of dominance. What exactly is dominance? Why are some alleles dominant and some are recessive? To tackle these questions, we must understand that what a gene does. Every gene, as you know by now, contains the information to express a particular trait. In a diploid organism, there are two copies of each gene, that is, as a pair of alleles. Now, these two alleles need not always be identical as in a heterozygote. One of them may be different due to some changes that it has undergone, about which you will read further on and in the next chapter, which modifies the information that particular allele contains. Let's take an example of a gene that contains the information for producing an enzyme. Now there are two copies of this gene, the two allelic forms. Let us assume, as is more common, that the normal allele produces the normal enzyme that is needed for the transformation of a substrate capital S. Theoretically, the modified allele could be responsible for production of the normal or less efficient enzyme or a non-functional enzyme or no enzyme at all. In the first case, the modified allele is equivalent to the unmodified allele. That is, it will produce the same phenotype or trait, that is, result in the transformation of substrate yes. Such equivalent allele pairs are very common, but if the allele produces a non-functional enzyme or no enzyme, the phenotype may be affected. The phenotype or trait will only be dependent on the functioning of the unmodified allele. The unmodified or functioning allele, which represents the original phenotype, is the dominant allele, and the modified allele is generally the recessive allele. Hence, in the example above, the recessive trait is seen due to non-functional enzyme or because no enzyme is produced. 4.2.2.2 Codominance. Till now we were discussing crosses where the F1 resemble either of the two parents, that is dominance, or was in between incomplete dominance. But in the case of codominance, the F1 generation resembles both the parents. A good example is different types of red blood cells that determine ABO blood grouping in human beings. ABO blood groups are controlled by the gene capital I. The plasma membrane of the red blood cells has sugar polymers that protrude from its surface, and the kind of sugar is controlled by the gene. The gene capital I has three alleles, IA, IB and small I. The alleles IA and IB produce a slightly different form of sugar, while allele small I does not produce any sugar. Because humans are diploid organisms, each person possesses any two of the three I gene alleles. IA and IB are completely dominant over small I. In other words, when IA and small I are present, only IA expresses because small I does not produce any sugar. And when IB and small I are present, IB expresses. But when IA and IB are present together, they both express their own types of sugars. This is because of codominance. Hence, red blood cells have both capital A and capital B types of sugars. Since there are three different alleles, there are 6 different combinations of these three alleles that are possible, and therefore a total of 6 different genotypes of the human ABO blood types. How many phenotypes are possible? Table 4.2 shows the genetic basis of blood groups in human population. Do you realize that the example of ABO blood grouping also provides a good example of multiple alleles? Here you can see that there are more than 2, that is 3 alleles governing the same character. Since in an individual only 2 alleles can be present, multiple alleles can be found only when population studies are made. Occasionally, a single gene product may produce more than one effect. For example, starch synthesis in pea seeds is controlled by one gene. It has 2 alleles, that is capital B and small b. Starch is synthesized effectively by capital B homozygotes, and therefore larger starch grains are produced. In contrast, small b homozygotes have lesser efficiency in starch synthesis and produce smaller starch grains. After maturation of seeds, capital B seeds are round and the small b seeds are wrinkled. Heterozygotes produce round seeds, and so capital B seems to be dominant allele. But the starch grains produced are of intermediate size in capital B seeds. So if starch grain size is considered as the phenotype, then from this angle the alleles show incomplete dominance. Therefore, dominance is not an autonomous feature of a gene or the product that it has information for. It depends as much on the gene product and the production of a particular phenotype from this product as it does on the particular phenotype that we choose to examine. In case more than one phenotype is influenced by the same gene. 4.3 Inheritance of 2 genes. Mendel also worked with a crossed pea plants that different in 2 characters, as is seen in the cross between a pea plant that has seeds with yellow color and round shape and one that had seeds of green color and wrinkled shape, as shown in figure 4.7. Mendel found that the seeds resulting from crossing of the parents had yellow colored and round shaped seeds. Here you can tell which of the character is in the pairs yellow or green color and round or wrinkled shape was dominant. Thus, yellow color was dominant over green and round shape dominant over wrinkled. These results were identical to those that he got when he made separate monohybrid crosses between yellow and green seeded plants and between round and wrinkled seeded plants. Let us use genotypic symbols capital Y for dominant yellow seed color and small y for recessive green seed color, capital R for round shaped seeds and small r for wrinkled seed shape. The genotype of the parents can then be written as capital R, capital Y, and small r, small y, small y. The cross between the two plants can be written down as in figure 4.7, showing the genotypes of the parent plants. The gametes capital R, capital Y and small r, small y unite on fertilization to produce the F1 hybrid capital R, capital Y, small y. When Mendel self-hybridized the F1 plants, he found that 3/4 of F2 plants had yellow seeds and 1/4 had green. The yellow and green color segregated in a 3:1 ratio. Round and wrinkled seed shape also segregated in a 3:1 ratio, just like in a monohybrid cross. 4.3.1 Law of Independent Assortment. In the dihybrid cross, the phenotypes round yellow, round green and wrinkled green appeared in the ratio 9:3:3:1. Such a ratio was observed for several pairs of characters that Mendel studied. The ratio of 9:3:3:1 can be derived as a combination series of 3 yellow:1 green with 3 round:1 wrinkled. This derivation can be written as follows. 3 round:1 wrinkled, 3 yellow:1 green is equal to 9 round yellow as to 3 wrinkled yellow as to 3 round green as to 1 wrinkled green. Based upon such observations on dihybrid crosses, that is crosses between plants differing in two traits, Mendel proposed a second set of generalization that we call Mendel's law of independent assortment. The law states that when two pairs of traits are combined in a hybrid, segregation of one pair of characters is independent of the other pair of characters. The Punnett square can be effectively used to understand the independent segregation of two pairs of genes during meiosis and the production of eggs and pollen in the F1, capital R, capital Y, small y plant. Consider the segregation of one pair of genes capital R and small r. 50% of gametes have the gene capital R and the other 50% have small r. Now, besides each gamete having either capital R or small r, it should also have the allele capital Y or small y. The important thing to remember here is that segregation of 50% capital R and 50% small r is independent from the segregation of 50% capital Y and 50% small y. Therefore, 50% of the small r bearing gametes has capital Y and the other 50% has small y. Similarly, 50% of the capital R bearing gametes has capital Y and the other 50% has small y. Thus, there are 4 genotypes of gametes, that is 4 types of pollen and 4 types of eggs. The 4 types of are capital R, capital Y, capital R, small r, capital Y and small r, small y, each with a frequency of 25% or 1/4 of the total gametes produced. When you write down the 4 types of eggs and pollen on the two sides of the Punnett square, it is very easy to derive the composition of the zygote that give rise to the F2 plant. 4.3.2 Chromosomal Theory of Inheritance. Mendel published his work on inheritance of characters in 1865, but for several reasons it remained unrecognized till 1900. Firstly, communication was not easy as it is now in those days, and his work could not be widely publicized. Secondly, his concept of genes or factors, in Mendel's words, as stable and discrete units that controlled the expression of traits and of the pair of alleles which did not blend with each other, was not accepted by his contemporaries as an explanation for the apparently continuous variations seen in nature. Thirdly, the Mendel's approach of using mathematics to explain biological phenomena was totally new and unacceptable to many of the biologists of his time. Finally, though Mendel's work suggested that the factors or genes were discrete units, he could not provide any physical proof for the existence of factors or say what they were made of. In 1900, three scientists De Vries, Correns, and von Tessemark independently rediscovered Mendel's result on the inheritance of characters. Also, by this time, due to advancements in microscopy that were taking place, scientists were able to carefully observe cell division. This led to the discovery of structure in the nucleus that appeared to double and divide just before each cell division. These were called chromosomes, colored bodies, as they were visualized by staining. By 1902, the chromosome movement during meiosis had been worked out. Walter Sutton and Theodore Bory noted that the behavior of chromosomes was parallel to the behavior of genes and used chromosome movement to explain Mendel's law. Recall that you have studied the behavior of chromosomes during mitosis, that is equational division, and during meiosis, that is reduction division. The important thing to remember are the chromosomes as well as genes occur in pairs. The two alleles of gene pair are located on homologous sites on homologous chromosomes. Figure 4.8 shows a meiosis and germ cell formation in a cell with 4 chromosomes. Can you see how chromosomes segregate when germ cells are formed? Table 4.3 shows a comparison between the behavior of chromosomes and genes. Column A and B both occur in pairs. A segregate at the time of gamete formation such that only one of each pair is transmitted to a gamete, where B segregate at gamete formation and only one of each pair is transmitted to a gamete. A is independent pairs segregate independently of each other, and B, one pair segregates independently of another pair. Can you tell which of these column A or B represent the chromosome and which represents the gene? How did you decide? During anaphase of meiosis first, the two chromosome pairs can align at the metaphase plate independently of each other. To understand this, compare the chromosomes of 4 different colors in a left and right columns. In left column, orange and green is segregating together, but in the right hand column, the orange chromosome is segregating with the red chromosomes. Figure 4.9 shows an independent assortment of chromosomes. Sutton and Bory argued that the pairing and separation of a pair of chromosomes would lead to the segregation of a pair of factors they carried. Sutton united the knowledge of chromosomal segregation with Mendelian principles and called it the chromosomal theory of inheritance. Following this synthesis of ideas, experimental verification of the chromosomal theory of inheritance by Thomas Hunt Morgan and his colleagues led to discovering the basis of the variation that sexual reproduction produce. Morgan worked with the tiny fruit flies, Drosophila melanogaster, which were found very suitable for such studies. They could be grown on simple synthetic medium in the laboratory. They complete their life cycle in about 2 weeks, and a single mating could produce a large number of progeny flies. Also, there was a clear differentiation of the sexes. The male and female flies are easily distinguishable. Also, it has many types of hereditary variations that can be seen with low power microscope. 4.3.3 Linkage and Recombination. Morgan carried out several dihybrid crosses in Drosophila to study genes that were sex-linked. The crosses were similar to the dihybrid crosses carried out by Mendel in peas. For example, Morgan hybridized yellow-bodied white-eyed females to brown-bodied red-eyed males and intercrossed their F1 progeny. He observed that the two genes did not segregate independently of each other, and the F2 ratio deviated very significantly from 9:3:3:1 ratio, expected when the two genes are independent. Morgan and his groups knew that the genes were located on X chromosome and saw quickly that when the two genes in a dihybrid cross were situated on the same chromosome, the proportion of the parental gene combinations were much higher than the non-parental type. Morgan attributed his due to physical association or linkage of the two genes and coined the term linkage to describe this physical association of genes on a chromosome, and the term recombination to describe the generation of non-parental gene combinations. Morgan and his group also found that even when genes were grouped on the same chromosome, some genes were very tightly linked while others were loosely linked. For example, he found that the genes white and yellow were very tightly linked and showed only 1.3% recombination, while white and miniature wing showed 37.2% recombination. His student Alfred Stewart Went used the frequency of recombination between gene pairs on the same chromosome as a measure of the distance between genes and mapped their position on the chromosome. Today, genetic maps are extensively used as a starting point in the sequencing of a whole genome, as was done in the case of human genome sequencing project described later. 4.4 Polygenic inheritance. Mendel's studies mainly described those traits that have distinct alternate forms such as flower color, which are either purple or white. But if you look around, you will find that there are many traits which are not so distinct in their occurrence and are spread across a gradient. For example, in humans, we don't just have tall or short people as two distinct alternatives, but a whole range of possible heights. Such traits are generally controlled by three or more genes and are thus called as polygenic traits. Besides the involvement of multiple genes, polygenic inheritance also takes into account the influence of environment. Human skin color is another classic example for this. In a polygenic trait, the phenotype reflects the contribution of each allele. That is, the effect of each allele is additive. To understand this better, let us assume that three genes capital A, B, C control skin color in human with the dominant forms capital A, B and C responsible for dark skin color and the recessive forms small a, B and C for light skin color. The genotype with all the dominant alleles capital AA, BB, CC will have the darkest skin color, and that all the recessive alleles small aa, bb, cc will have the lightest skin color. As expected, the genotype with the three dominant alleles and three recessive alleles will have an intermediate skin color. In this manner, the number of each type of alleles in the genotype would determine the darkness or lightness of the skin in an individual. 4.5 Pleiotropy. We have so far seen the effect of a gene on a single phenotype or trait. There are, however, instances where a single gene can exhibit multiple phenotypic expression. Such a gene is called a pleiotropic gene. The underlying mechanism of pleiotropy in most cases is the effect of gene on metabolic pathway which contribute towards different phenotypes. An example of this is the disease phenylketonuria, which occurs in humans. The disease is caused by mutation in the gene that codes for the enzyme phenylalanine hydroxylase, that is single gene mutation. This manifests itself through phenotypic expression characterized by mental retardation and a reduction in hair and skin pigmentation. 4.6 Sex determination. The mechanism of sex determination has always been a puzzle before the geneticists. The initial clue about the genetic or chromosomal mechanism of sex determination can be traced back to some of the experiments carried out in insects. In fact, the cytological observations made in a number of insects led to the development of the concept of genetic or chromosomal basis of sex determination. Henking in 1891 could trace a specific nuclear structure all through spermatogenesis in a few insects, and it was also observed by him that 50% of the sperms received this structure after spermatogenesis, whereas the 50% sperm did not receive it. Henking gave a name to this structure as the X body, but he could not explain its significance. Further investigations by other scientists led to the conclusion that the X body of Henking's was in fact a chromosome, and that is why was given the name X chromosome. It was also observed that in a large number of insects, the mechanism of sex determination is of XO type. That is, all eggs bear an additional X chromosome besides the other chromosomes, that is autosome. On the other hand, some of the sperms bear the X chromosome whereas some do not. Eggs fertilized by sperm having an X chromosome become female, and those fertilized by sperms that do not have an X chromosome become males. Do you think the number of chromosomes in the male and female are equal? Due to the involvement of the X chromosome in the determination of sex, it was designated to be the sex chromosome, and the rest of the chromosomes were named as autosomes. Grasshopper is an example of XO type or of sex determination in which the males have only one X chromosome besides the autosomes. whereas females have a pair of X chromosomes. These observations led to the investigation of a number of species to understand the mechanism of sex determination. In a number of other insects and mammals, including man, XY type of sex determination is seen where both male and female have same number of chromosomes. Among the male, and X chromosome is present, but its counterpart is distinctly smaller and called the Y chromosome. Females, however, have a pair of X chromosome. Both males and females bear same number of autosomes. Hence, the males have autosomes plus XY while females have autosomes plus XX. In human beings and in Drosophila, the males have one X and one Y chromosome, whereas females have a pair of X chromosomes besides autosomes. In the above description, you have studied about two types of sex determining mechanisms, that is XO type and XY type. But in both cases, males produce two different types of gametes, A, either with or without X chromosome, or B, some gametes with X chromosome and some with Y chromosome. Such types of sex determination mechanism is designated to be the example of male heterogamety. In some other organisms, for example birds, a different mechanism of sex determination is observed. In this case, the total number of chromosomes is same in both males and females, but two different types of gametes in terms of the sex chromosome are produced by females, that is female heterogamety. In order to have a distinction with the mechanism of sex determination described earlier, the two different sex chromosomes of a female bird has been designated to be the Z and W chromosomes. In these organisms, the females have one Z and one W chromosome, whereas males have a pair of Z chromosomes besides the autosomes. 4.6.1 Sex determination in humans. It has already been mentioned that the sex determining mechanism in case of human is XY type. Out of 23 pairs of chromosomes present, 22 pairs are exactly same in both males and females. These are the autosomes. A pair of X chromosome are present in the female, whereas the presence of X and Y chromosome are determinant of the male characteristics. During spermatogenesis among males, two types of gametes are produced. 50% of the total sperm produced carry the X chromosome and the rest 50% has Y chromosome besides the autosomes. Females, however, produce only one type of ovum with an X chromosome. There is an equal probability of fertilization of the ovum with the sperm carrying either the X or Y chromosome. In case of ovum fertilizes with a sperm carrying X chromosome, the zygote develops into a female XX and the fertilization of ovum with Y chromosome carrying sperm results into a male offspring. Thus, it is evident that it is the genetic makeup of the sperm and determines the sex of the child. It is also evident that in each pregnancy there is always 50% probability of either a male or female child. It is unfortunate that in our society women are blamed for giving birth to female children and have seen ostracized and ill-treated because of this false notion. 4.6.2 Sex determination in honeybee. The sex determination in honeybee is based on the number of sets of chromosomes an individual receives. An offspring formed from union of the sperm and the eggs develops as a female, that is queen or worker, and an unfertilized egg develops as a male, that is drawn by means of parthenogenesis. This means that the males have half the number of chromosomes than that of the female. The females are diploid having 32 chromosomes and males are haploid, that is giving 16 chromosomes. This is called a haplodiploid sex determination system and has special characteristics features such as the male produces sperms by mitosis. They do not have father and thus cannot have sons, but have a grandfather and can have grandsons. How is the sex determination mechanism different in the birds? Is the sperm or the egg responsible for the sex of the chicks? 4.7 Mutation. Mutation is a phenomenon which results in the alteration of DNA sequences and consequently results in the changes in genotype and the phenotype of an organism. In addition to the recombination, mutation is another phenomenon that leads to variation in DNA. As you learn in chapter 5, one DNA helix runs continuously from one end to other in each chromatid in a highly supercoiled form. Therefore, loss, that is deletion or gain, insertion or duplication of a segment of DNA result in alteration in chromosomes. Since genes are known to be located on chromosomes, alteration in chromosomes results in abnormalities or aberrations. Chromosomal aberrations are commonly observed in cancer cells. In addition to the above, mutation also arise due to change in a single base pair of a DNA. This is known as point mutation. A classical example of such mutation is sickle cell anemia. Deletion and insertions of base pairs of DNA causes frame shift mutations. The mechanism of mutation is beyond the scope of this discussion at this level. However, there are many chemical and physical factors that induce mutations. These are referred to as mutagens. UV radiations can cause mutations in organisms. It is a mutation. 4.8 Genetic disorders. 4.8.1 Pedigree analysis. The idea that disorders are inherited has been prevailing in the human society since long. This was based on the heritability of certain characteristic features in families. After the rediscovery of Mendel's work, the practice of analyzing inheritance pattern of traits in human beings began. Since it is evident that control crosses that can be performed in pea plant or some other organisms are not possible in case of human beings. Study of the family history about inheritance of a particular trait provides an alternative. Such an analysis of trait in a several generations of a family is called a pedigree analysis. In the pedigree analysis, the inheritance of a particular trait is represented in the family tree over generation. In human genetics, pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait, abnormality, or disease. Some of the important standard symbols used in the pedigree analysis have been shown in figure 4.13. As you have studied in this chapter, each and every feature in any organism is controlled by one or other gene located on the DNA present in the chromosome. DNA is the carrier of genetic information. It is hence transmitted from one generation to the other without any change or alteration. However, changes or alteration do take place occasionally. Such an alteration or changes in the genetic material is referred to as mutation. A number of disorders in human beings have been found to be associated with the inheritance of changed or altered genes or chromosomes. 4.8.2 Mendelian disorders. Broadly, genetic disorders may be grouped into two categories: Mendelian disorders and chromosomal disorders. Mendelian disorders are mainly determined by alteration or mutation in single gene. These disorders are transmitted to the offspring on the same lines as we have studied in principle of inheritance. The pattern of inheritance of such Mendelian disorder can be traced in a family by the pedigree analysis. Most common and prevalent Mendelian disorder are hemophilia, cystic fibrosis, sickle cell anemia, color blindness, phenylketonuria, thalassemia, etc. It is important to mention here that such Mendelian disorders may be dominant or recessive. By pedigree analysis, one can easily understand whether the trait is in question is dominant or recessive. Similarly, the trait may also linked to the sex chromosome as in case of hemophilia. It is evident that this X-linked recessive trait shows transmission from carrier female to male progeny. A representative pedigree is shown in figure 4.14. For dominant and recessive traits, discuss with your teacher and design pedigree for characters linked to both autosomes and sex chromosome. Color blindness. It is a sex-linked recessive disorder due to defect in either red or green cone of eye resulting in failure to discriminate between red and green color. This defect is due to mutation in certain genes present in the X chromosome. It occurs in about 8% of males and only about 0.4% of females. This is because the genes that lead to red-green color blindness are on the X chromosome. Males have only one X chromosome and females have two. The son of the woman who carries the gene has a 50% chance of being colorblind. The mother is not herself colorblind because the gene is recessive. That means that its effect is suppressed by her matching dominant normal gene. A daughter will not normally be colorblind unless her mother is a carrier and her father is colorblind. Hemophilia. This sex-linked recessive disease, which shows its transmission from unaffected carrier female to some of the male progeny, has been widely studied. In this disease, a single protein that is a part of the cascade of proteins involved in the clotting of blood is affected. Due to this, in an affected individual, a simple cut will result in non-stop bleeding. The heterozygous female that is carrier for hemophilia may transmit the disease to sons. The possibility of a female becoming a hemophilic is extremely rare because mother of such a female has to be at least a carrier and the father should be hemophilic. The family pedigree of Queen Victoria shows a number of hemophilic descendants as she was carrier of the disease. Sickle cell anemia. This is an autosome-linked recessive trait that can be transmitted from parents to the offspring when both the partners are carrier for the gene or heterozygous. The disease is controlled by a single pair of allele, that is hemoglobin A and hemoglobin S, that is HbA and HbS. Out of the three possible genotypes, only homozygous individuals for HbS, that is HbS, show the diseased phenotype. Heterozygous, that is HbA, HbS individuals appear apparently unaffected, but they are carrier of the disease as there is 50% probability of transmission of mutant gene to the progeny, thus exhibiting sickle cell trait as shown in figure 4.15. The defect is caused by a substitution of glutamic acid by valine at the 6th position of beta globin chain of the hemoglobin molecule. The substitution of amino acid in the globin protein results due to single base substitution at the 6th codon of the beta globin gene from GAG to GUG. The mutant hemoglobin molecule undergoes polymerization under low oxygen tension, causing the change in the shape of the RBC from biconcave disc to elongated sickle-like structure. Phenylketonuria. This inborn error of metabolism is also inherited as the autosomal recessive trait. The affected individual lacks an enzyme that converts the amino acid phenylalanine into tyrosine. As a result of this phenylalanine is accumulated and converted into phenylpyruvic acid and other derivatives. Accumulation of these in brain results in mental retardation. These are also exerted through urine because of its poor absorption by kidney. Thalassemia. This is also an autosome-linked recessive blood disease transmitted from parents to the offspring when both the partners are unaffected carrier for the gene or heterozygous. The defect could be due to either mutation or deletion, which ultimately results in reduced rate of synthesis of one of the globin chains, that is alpha and beta chains, make up the hemoglobin. This causes the formation of abnormal hemoglobin molecules resulting into anemia, which is characteristic of the disease. Thalassemia can be classified according to which chain of the hemoglobin molecule is affected. In alpha thalassemia, production of alpha globin chain is affected, while in beta thalassemia, production of globin chain is affected. Alpha thalassemia is controlled by two closely linked genes, that is HbA1 and HbA2 on chromosome 16 of each parent, and it is observed due to mutation or deletion of one or more of the four genes. The more genes affected, the less alpha globin molecules produced, while beta thalassemia is controlled by a single gene HBB on chromosome 11 of each parent and occurs due to mutation of one or both the genes. Thalassemia differs from sickle cell anemia in that the former is a quantitative problem of synthesizing too few globin molecules, while the latter is a qualitative problem of synthesizing an incorrectly functioning globin. 4.8.3 Chromosomal disorders. The chromosomal disorders, on the other hand, are caused due to absence or excess or abnormal arrangement of one or more chromosomes. Failure of segregation of chromatids during cell division cycle results in the gain or loss of chromosomes called aneuploidy. For example, Down syndrome results in the gain of extra copy of chromosome 21. Similarly, Turner syndrome results due to loss of an X chromosome in human females. Failure of cytokinesis after telophase stage of cell division results in an increase in a whole set of chromosomes in an organism, and this phenomena is known as polyploidy. This condition is often seen in plants. The total number of chromosomes in a normal human cell is 46, that is 23 pairs. Out of these, 22 pairs are autosomes and one pair of chromosomes are sex chromosome. Sometimes Though rarely, either an additional copy of chromosome may be included in an individual or an individual may lack one of any one pair of chromosomes. These situations are known as trisomy or monosomy of a chromosome respectively. Such a situation leads to a very serious consequences in individual. Down syndrome, Turner syndrome, Klinefelter syndrome are common examples of chromosomal disorders. Down syndrome. The cause of this genetic disorder is the presence of an additional copy of the chromosome number 21, that is trisomy of 21. This disorder was first described by Langdon Down in 1866. The affected individual is short statured with small round head, furrowed tongue and partially open mouth. Palm is broad with characteristic palm crease. Physical, psychomotor and mental development is retarded. Klinefelter syndrome. This genetic disorder is also caused due to the presence of an additional copy of X chromosome, resulting into a karyotype 47, that is XXY. Such an individual has overall masculine development. However, the feminine development, that is development of breast, that is gynecomastia, is also expressed. Such individuals are sterile. Turner syndrome. Such a disorder is caused due to the absence of one X chromosome, that is 45 with XO. Such females are sterile as ovaries are rudimentary besides other features including lack of other secondary sexual characters. Let us discuss the summary of the chapter Principles of Inheritance and Variation. Genetics is a branch of biology which deals with the principle of inheritance and its practices. Progeny resembling the parents in morphological and psychological features has attracted the attention of many biologists. Mendel was the first to study this phenomena systematically. While studying the pattern of inheritance in pea plants of contrasting characters, Mendel proposed the principles of inheritance, which are today referred to as Mendel's law of inheritance. He proposed that the factors, that is, later named as genes regulating the characters, are found in pairs known as alleles. He observed that the expression of characters in the offspring follow a definite pattern in different first generations, that is F1, second, F2, and so on. Some characters are dominant over others. The dominant characters are expressed when factors are in heterozygous condition, that is law of dominance. The recessive characters are only expressed in homozygous conditions. The characters never blend in heterozygous condition. A recessive character that was not expressed in heterozygous condition may be expressed again when it becomes homozygous. Hence, characters segregate while formation of gametes, that is law of segregation. Not all characters show true dominance. Some characters show incomplete and some show codominance. When Mendel studied the inheritance of two characters together, it was found that the factors independently assort and combine in all permutations and combinations, that is law of independent assortment. Different combinations of gametes are theoretically represented in a square tabular form known as Punnett square. The factors, that is now known as gene on chromosomes regulating the characters, are called the genotype, and physical expression of the character is called the phenotype. After knowing that the genes are located on the chromosome, a good correlation was drawn between Mendel's law, that is segregation and assortment of chromosomes during meiosis. The Mendel's laws were extended in the form of chromosomal theory of inheritance. Later, it was found that Mendel's law of independent assortment does not hold true for the genes that were located on the same chromosomes. These genes were called as linked genes. Closely located genes assorted together, the distantly located genes, due to recombination, assorted independently. Linkage maps, therefore, corresponded to arrangements of genes on a chromosome. Many genes were linked to sexes also and called as sex-linked genes. The two sexes, that is male and female, were found to have a set of chromosomes which were common and another set was different. The chromosomes which were different in two sexes were named as sex chromosomes. The remaining set was named as autosomes. In humans, a normal female has 22 pairs of autosomes and a pair of sex chromosomes XX. A male has 22 pairs of autosomes and a pair of sex chromosomes as XY. In chicken, sex chromosome in male are ZZ and in female are ZW. Mutation is defined as change in the genetic material. A point mutation is change of a single base pair in DNA. Sickle cell anemia is caused due to change of one base in the gene coding for beta chain of hemoglobin. Inheritable mutations can be studied by generating a pedigree of family. Some mutations involve changes in whole set of chromosomes, that is polyploidy, or change in a subset of chromosome number, that is aneuploidy. This helped in understanding the mutational basis of genetic disorders. Down syndrome is due to trisomy of chromosome 21, where there is an extra copy of chromosome 21 and consequently the total number of chromosomes becomes 47. In Turner syndrome, one X chromosome is missing and the sex chromosome is as XO. And in Klinefelter syndrome, the condition is XXY. These can be easily studied by analysis of karyotypes. Thank you for watching this video. Please subscribe and press the bell icon for further videos.